You are a physicist, you're a computer scientist, you're a biologist, you are like a package all in one. What do you call yourself?
I'm a microscopist. I like to build microscopes, and I'm passionate about applying them to image biology.
What is that thing that intrigued in you in the small creatures?
The entire worm is only the size of about a comma in a sentence, and yet it has behaviors. It has brain, it has learning and memory. And so it kind of intrigued me. How does this simple organism work? The wonderful thing about exploring the microworld, chances are very, very good you will see something that nobody has ever seen before. Pay attention to problems, because problems and failures are the wheel that drive progress. And very often, frustration in science are really opportunities in disguise.
Good evening, Hari, and welcome to Ashoka University. Thank you for coming here. You are a physicist, you're a computer scientist, you're a biologist, you are like a package all in one. What do you call yourself?
Right. Well, first of all, thanks for the opportunity to participate. It's really fun to have this conversation with you and to come to Ashoka University. My bread and butter is sort of taking tools from physics and engineering and building tools to inform biologists about things that they can see. And so, if I had to call myself something, I guess I would say I'm a microscopist.
Right.
I like to build microscopes, and I'm passionate about applying them to image biology.
Right. And you work in Janelia Research Campus. So I think this is something people would be curious to know also. What is the campus about?
Yeah, thanks. So Janelia Research Campus is part of actually a bigger umbrella organization called the Howard Hughes Medical Institute. And this is I think the second biggest nonprofit biomedical funder after the Wellcome Trust. And so in the early 2000s, things were going well for the Howard Hughes Medical Institute, HHMI, as it's called. And they mostly funded labs across the United States. So they would give grants to researchers, but their endowment was doing well. And they had a decision to make: either fund twice as many scientists at universities, or they could find another use for their money. And so they decided to make this research campus called Janelia to sort of like have like a biological Bell Labs environment. So this is a campus focused on scientific biomedical research to try to tackle problems that were kind of outside the bounds of conventional funding.
Right.
And so this was born in sort of 2005, 2006. Actually started out as a napkin on a piece of paper, a napkin even before that. But then the building was finished in 2006, and I was one of the first employees. Now, in answer to your question, why Janelia?
Yeah.
So the property is on, I think, something like a few hundred acres in the state of Virginia, about an hour outside of DC. And it's actually purchased on some farmland, some historical farmland owned by a farmer. And the story goes, he had two daughters, Jane and Cornelia.
Mm-hmm.
And so the deal was anybody that wanted to buy this land had to maintain the name Janelia, which is a fusion of Jane and Cornelia. So that's why we have this funny name. And it used to actually be called Janelia Farm Research Campus. Along the way, the farm part got dropped off.
Right. No, that's interesting. So in this research campus, you have a lot of physicists, biologists, and computer scientists all working in one go. And how does your day look like working in that place?
Yeah, exactly right. So a couple of unique things about Janelia. It was designed to be a place where biologists could work hand in hand with tool builders, which isn't always easy in a conventional academic setting.
Right.
And so one of the great things about this place is that although it's small, we have less than 1,000 people on the campus. It's very, everybody is in one building. So there really is this high frequency of collisional interaction between physicists and biologists. And so another unique thing about Janelia is that you're not allowed to write grants. So all of the money is internally funded by HHMI, and the group leaders, I would be considered a group leader. We have small labs. My lab size is capped at 6, and we have to work in the lab. That's kind of a requirement.
Yeah.
So unlike, maybe having a larger lab at a university where I'd be spending time doing administration, writing grants, here I have a chance to still actually work with my hands and build things which is really fun.
Many people do not know what is dispim and isim. So if you have to sort of explain that to a lay audience, how would you explain that?
Sure. So, we're in the business of imaging very small biological critters and
Right.
creatures like single cells or like small embryos from different model organisms like
Right.
worms or fish. And one of the challenges when you image these specimens is that they're sensitive to light.
Yeah.
So just like we would get sunburned if we spent too much time outside in the hot Delhi sun, so too with these small biological organisms. If you shine too much light on them, then they die.
Yeah, you
Their
fry
cells-
them.
You fry them. You
Yeah.
fry them. And so that introduces all kinds of interesting trade-offs for the microscopist, right? The challenge is, how do you keep the thing alive when, in order to image it, you have to shine light on it? And so there are various ways in which we can use less light. In fact, one of the ideas is we use, instead of illuminating the entire specimen at once, we illuminate only the part that we look at
Yeah.
with a thin light sheet. And this idea actually goes back more than 100 years to some German researchers. It was kind of rediscovered in the 2000s by a whole bunch of groups. And so this dispim that you mentioned is a particular implementation of this idea that is very well suited to studying small things like embryos with pretty high resolution in all three dimensions without killing it.
Right. So, and this is all by the fluorescence properties of
Yes.
the proteins that you put in them
Exactly. Not
Yeah.
just proteins. but
and dyes.
And dyes. You make
Yeah.
a good point. So my kind of playground is in a subset of microscopy called fluorescence microscopy,
Yeah.
meaning in order to like see something, we have to label it with a fluorescent dye. And so
Yeah.
we might illuminate the specimen with a laser. That laser causes these dye molecules to glow, and then we capture the fluorescence. So you're right. The dispim and the isim are kinds of fluorescence microscopes.
Right. So that's interesting that you are primarily an instrumentation person, and then you went on to look at biological samples, right? So
That's right.
what is that thing that intrigued in you in the small creatures? Is it their beauty, or is it the shapes at a microscopy level? Is it a pattern? What is it?
Right. So yeah, you're right. My bread and butter is microscopy. And when I was actually finishing at Janelia, I was trying to wrestle with my future. And I was trying to think, should I just build tools or should I try to, apply these tools to biology? What is the right balance
Yeah.
for me? And, my heart will always be in tool building. I think that's just the way that my brain is wired. But when I was on the job market, one of the other places I was considering was Yale University. And when I was interviewing at Yale, I met this young neuroscientist, Daniel Colin-Ramos, who had just started his lab at Yale. And he was like, well, you have these great microscopes. You have these great ideas for imaging with light sheet microscopy. I have the perfect biological problem, which is studying how the brain of these simple worms, C. elegans, form. I mean, this is something we can't do very well because of this light dose, because of the phototoxicity,
Yeah.
and because we need very high resolution. And so your technologies would be perfect. And so that was actually my introduction to the worm. It was quite by accident. But as I learned more and more about the worm, it also appealed to me on kind of a deeper level than just studying its brain. This worm, C. elegans, is a very famous model organism. And the reason it's famous is in part due to its kind of beautiful simplicity. It has only 302 neurons in the adult worm. The entire worm is only the size of about a comma in a sentence, so you can barely see it with the naked eye. It's a very simple organism, and yet it has behaviors. It has a brain. It has learning and memory. And so it kind of intrigued me. How does this simple organism work? And that has kind of motivated me throughout the years.
Right. So now there's a lot of buzz around AI, right? So do you use a lot of AI in your, tracking the systems or tracking the brain development, or do you integrate these functionalities in there?
We do a bit. So one of the challenges in microscopy is this pyramid of frustration. So if you try to make a microscope see something smaller,
Yeah.
then you typically have to shine more light on it. We would say that in order to improve spatial resolution, we sacrifice the health of the sample. This is
Yeah.
one classic trade-off. Another one might be if we try to image something faster, if we try to record at a higher frame rate, then there is less signal in each frame. So we would say the signal to noise ratio is reduced when imaging it fast. So microscopy is rife with these trade-offs, which makes my life interesting. And with instrumentation alone, one is limited by physics. Okay. So the reason we use deep learning is that, normally when we use microscopes, our attitude is like, okay, the microscope is a discovery tool. How can we use it to see something we've never seen before? And I think this is the main value of a microscope. But you can kind of turn this idea on its head. You could ask, how do previous experiments inform future experiments? So if I've imaged a worm embryo 1,000 times, right? I've imaged what all of its little parts look like. Maybe that can inform a future experiment. Maybe I can use that prior knowledge to sharpen up an image or to denoise an image. And this is where neural networks and AI can be useful, because they give one a way of using prior information to make the images sharper, or make the images higher signal to noise ratio given this prior. But I do want to say that there's one big caveat with all of these AI methods, including the ones you may have heard about like ChatGPT or the methods that I use. And that is that they're trained on data. And so they are subject to the laws of the data. If they see something that is outside of the training data, they can hallucinate. And this is something you may have heard of. It's well known in large language models.
Right.
So we do have to be careful when we use these AI tools, but they're kind of a fantastic new tool to have. And, like any tool, one needs to know the strengths and the weaknesses as one adopts the tool.
Right. So, the fluorescence microscopy has evolved over time, just by the techniques, the inventions there at that bit, but also with integration of AI and computer science and all those kind of algorithms. I think it has gone now far, at a much greater speed. So now if you look into the future, what do you see? How do you think that the field of, let's say, fluorescence microscopy is going to evolve?
Yeah, great question. So I think since you mentioned AI, one thing that I think in the future is we'll see more and more automation, right? So, we're used to still today manually tuning the knobs in the microscope. And, there's something that I like about that, because if you understand what you're doing, you understand the microscope. But many of these adjustments are based on our experience, right? Our kind of feel for the sample. And I think for many samples, I could imagine a future where this is more and more automated by AIs, right? So the computer will decide, okay, if you want to image this sample for so many frames, set the intensity like this, right? So there may be more of that. Another area that I think we'll see more and more sort of computer integration is the analysis of these images. So with the click of a button, one can generate terabytes and terabytes of data and petabytes of data even, right? And our ability to make sense of that data has not kept up with the sheer flood of the data that is coming. And part of that is because, our brains, they think in kind of 2 dimensions, right? Or maybe 2 dimensions over time. It's hard for us to think in 4 dimensions, right? Volumes over time. These computer programs are not bound by like some of the limitations of our brain. And so if we're trying to pull out patterns from these large data sets, I think we will see more and more AI models or non-AI models, even classical models from physics that pull out patterns from image data. I think we will see more of this as well.
When you design, certain methods of looking at things, you play with light. There are other fields also, like astronomy, for example, which is very closely related to microscopy, though the scales are very different. But then do you also take into consideration the problems there that could be solved by, inventing these methods? So do you do it?
Definitely, yeah. So I try to keep an eye always on related fields, and astronomy is a good example because, as you say, in the end, you're imaging things, you're imaging stars, you're imaging molecules, but the laws of optics are the same. Now, there are some important differences, but one thing that is similar is that in microscopy, when you look deep inside a sample, even though many of the samples we look at are transparent, they're not perfectly transparent. So the light that you put in or the light that you collect is kind of fuzzed out by the bending of light in the sample itself. And so that means that your images, especially when you look deep, are not as sharp, are not as finely detailed as the microscopy optics allow.
Yeah.
There's a very similar problem in astronomy. So if you try to take a picture of Saturn in a ground-based telescope, I mean, you can see the planet, but the image is not as sharp as in principle it could be given the optics of the telescope. And in astronomy, the problem is the variations in refractive index in the air create these sort of disturbances, right? And so the light on its way from the planet or the star or whatever gets bent in undesirable ways by the turbulence in the atmosphere. The astronomers figured out a way decades ago, really now, where if you could measure these disturbances, you could cancel them by applying equal and opposite disturbances. This is called adaptive optics in the parlance of astronomy. And my group, along with other groups, is trying to take ideas that have been deployed in astronomy and we're trying to port it over to microscopy. So this is one area where I think we will see more growth in the coming years because, you know, even today, most commercial microscopes only image the very superficial layers of a sample well.
Right.
When you go deeper and deeper in, it gets much harder to get a clear image. And so I think there's still room for technical innovation, for creativity in deep imaging. The other thing that that happens in microscopy, but probably in most fields, is that you develop something new. In this case, the new microscope, and that works pretty well. You can see more than you could than before, but no microscope is omniscient. There are always barriers. No microscope image is perfect. And so pretty soon after you develop something new, you start to find problems. You start to poke holes in the technology. And this led to kind of another idea and an improved version of this we called the dye spin, which provided even better resolution. So Ambika, the general thing, I think the general piece of advice would be pay attention to problems, because problems and failures are the wheel that drive progress. And very often, frustration in science are really opportunities in disguise.
Right.
And I've seen this many, many times in my career. I think, what is it that I'd really like to do that I just can't do? And, many times I don't have an answer, but sometimes I do have an answer. And that's often where there's room for innovation.
Right. So coming to that, because what people see is actually the tip of the iceberg that you usually show in your presentations. You have this, fabulous techniques. Everything is working super great.
Right.
But then how many times have you failed before that? And when it comes, can you give me an example where you were so sure about that this is what I want to work towards it? It didn't work out. Then what did you do?
I think so the only reason we have succeeded as we have is because we have failed more than other people.
Yeah.
If there's any secret to our success, it's failing and then really thinking about the failure, learning from the failure. And then we think about how to improve on what we did before. So there's this saying by, I think, Samuel Beckett, fail again, fail better. And that's what we try to do. This isn't necessarily fun. Nobody likes to fail, but it is important to kind of, to make the next success, right? In the case of the light sheet microscopes, the dye spims, we started building this. And, initially we didn't know what we were doing. I mean, we had never built a light sheet microscope. So, we probably killed many, many embryos in just setting up the microscope. And then we started to collect data, and we're fortunate to have collaborators who had looked at these embryos before with existing microscopes, and our images weren't quite as good. And so this led us to think, well, why aren't they as good? How do we tweak the microscope to make them better? And I think the key thing is we didn't stop. We didn't get discouraged. It's not a failure if you pick yourself up, right? And it's only really a failure if you stay down. And you might come to a point, right, in any particular project where you have to stop, and then you abandon it and you do something else. But a lot of times I think we succeed because we encounter some difficulty and we think and we keep thinking. And again, these failures are often opportunities. They're opportunities to learn, about why the thing isn't working, right? Which is, I think, important for science. This is how science progresses.
Yeah, and I think that's a very important lesson for all the people out there who is, planning to build things. That most often it fails.
Often it fails.
Yeah, it doesn't,
exactly
get there in the first go. But then people do not know when to give up and when to stop, when to start another thing, or how to rise from that failure. I think that's a big.
That's a
thing.
skill.
Yeah, that's a skill.
Yeah, it's not obvious sometimes, right? I mean, like if you're hammering your head against some problem, sometimes the best thing you can do is put that problem away for a little bit and come back. And that's maybe another point, right? So for the people that are listening to this, you might have ideas for scientific projects or in whatever field you're studying. And it may be that you don't have a line of attack, but the idea is still really a good idea. And so what I would say to you is remember these ideas, write them down. I have a book of unfinished ideas that I periodically look at, because you just never know when somebody else will have some tool that they make that will make your idea feasible, right? We're all standing on each other's shoulders. And so, don't get discouraged, right? I mean, you're kind of in this for the long game.
Right.
If you stay in science, it's kind of a marathon, not a sprint. So that might be another message, I think.
Right. So when you started off, Hari, it was like way early in your life. Like I think you made your first microscope when you were 16, 15?
Maybe a little bit older than that. Yeah, I made my first instrument when I was 16.
When you were 16, yeah.
Yeah.
So how did you even think about it? Because a child would think about, I don't know, playing with toys or something else, and maybe looking at his homework at school. But
Right.
then at that point, you were actually trying to build something. So what led to that?
Right. So maybe the backstory here is that I was very fortunate growing up in that I was able to skip high school basically and enroll in college early, because there happened to be a very good program at University of Washington where we could do this.
Oh.
And this was like the right thing for me, right? I mean, it was my I feel like my academic needs were not being met in middle school or high school. So I enrolled in this thing. And so I had an opportunity to work in a lab for this interesting man, a chemist who was interested in developing a method for studying pressure on the wings of honeybees, which sounds like a bizarre thing, which is kind of odd. But in any case, it was my job to figure out if this paint, this kind of phosphorescent sensor, was sensitive enough. So I think in this particular case, Ambika, the professor that I worked for, Jim Callis, had this idea for characterizing the paint using a very simple instrument. And I had learned enough physics that I knew about the physics of resonance, the physics of waves. And so I was able to put this theoretical idea that I'd learned about in my classes to make a functioning instrument that allowed me to fire sound waves at the paint and characterize it based on something I'd learned about in class. So this was kind of in some ways, a very lucky opportunity, because I got to work in this guy's lab who gave me enough freedom to try these things. And I was able to actually put what I had learned in my classes, in my physics classes, to use, which was really great. I mean, that probably is what inspired me for some of the fun of academic research, right? Sort of going into the unknown, but yet using ideas, it wasn't totally unknown, right? We had a handle on this problem. And so that's kind of how that particular thing happened.
So when did you start working with Eric?
Okay, so I worked with Eric Betzig as a postdoc. So in between, I went to graduate school at UC Berkeley. And at Berkeley, that's actually when I built my first microscope. I was trying to design a way of measuring forces. And so the scheme I came up with needed a microscope. And there was no commercial microscope again. So it was another case of like necessity being the mother of invention. There were some home-built instruments, so you could read about them in the literature. So I kind of read these papers, and by studying the papers, I was able to hack together a microscope, which I tailored for my use.
Right.
So that was sort of my first experience. And then towards the end of my PhD, when I was wondering what to do next, I was pretty sure I didn't want to do what many of my friends were doing, which were going to well-known labs, getting these fancy papers. I knew that I liked instrumentation, and I knew that I liked microscopes. I liked imaging. I also knew that like a big problem in imaging was the resolution limit.
Yeah.
And so it was again like some combination of my mind being ready and the right lucky opportunity presenting itself that Eric Betzig came to Berkeley, said he had this great idea for super resolution microscopy, and was anybody interested? So I went up to him after his talk and I said, I'm interested. I'm looking for a postdoc, and luckily he hired me, and I was able to work in his lab on this idea.
Okay. And he won the Nobel Prize.
He won the Nobel Prize. So he had this great idea for beating the diffraction limit. And I was lucky enough to work in his lab on implementing the idea, showing it was useful for biology. And I was one of the early, people that showed this concept could work in practice, which is something I'll always remember. And something very valuable. I also learned a lot of optics in that time, which kind of was very useful when I built my own lab and build more microscopes.
Right. So when Eric won the Nobel Prize, I think, when someone wins a Nobel Prize, it's actually a collective effort of many, right? Like there are many people working
Right.
on different aspects. Have you tried having conversations with artists somewhere across the world to understand these processes and communicate and work? Because I think there can be a lot of idea generation when you do that art and science mix.
That's a great idea. Yeah. So when I was at NIH, I was fortunate that in the neuroscience institutes, there was a resident artist. And so I did have some conversations with her about C. elegans, these worms and how they form, which she was
Right.
fascinated by. But, I think you're absolutely right. There could be more interaction if there were more opportunities for scientists and artists to meet. In fact, when I was preparing for this visit to Ashoka University,
Mhm.
somebody was kind enough to share Mahadevan's lecture
Yeah.
with me. And he actually talked about, many of his ideas are inspired by, I think the topic he was talking about was folding and origami,
Yes.
right?
Origami.
Which is this
Yes.
idea that has been around for a long time, right?
Yeah.
So he was inspired by that. So that would be one example that sort of comes to mind. And I think there could be more, right? I mean, artists see the world a very different way.
And on that note, we shift gears a little bit and get to some tricky, quick questions, which could be quite interesting also. So the first question I want to ask you is, if you were not a microscopist, what would you be?
Yeah. So I think I was talking about this with some students before this podcast. And
Yeah.
I think if I had to do it all over again, I would think a lot about teaching
Mm.
because I like it, even though I don't do very much of it. And I think particularly teachers at the very junior level, let's say elementary school, they have an amplifying effect on society, which is totally undervalued, right?
Yeah.
I mean, we don't know how many Feynmans or Einsteins
Right.
there are, but they're all there, in the population. And, we have to nurture these kind of talents, and good teaching can make these talents blossom, and bad teaching can destroy it.
Right.
So I think like if you wanted to do something with tremendous impact, I would consider something in that vocation.
Right.
One other thing I'll say about that is I think there's a tremendous opportunity to bridge the gap, the like political gap, right? So many of the problems we're facing in the United States with science are because we scientists haven't done a good enough job teaching the public about what we do,
Right.
what the value of science is. And many of us that are scientists, we know it, but the challenge is convincing the people that don't know it what the value is. And so I might think about that as well.
Okay, that's a very good answer. And is there any book or film that you would recommend to, younger generation or older generation even?
Wow. I mean, there are many, many books that I enjoyed. One book, so one of the things I like to try to study is the history of science, right? So at an institutional level, what makes a Bell Labs? What makes a Cambridge MRC, LMB, where there was a ton of innovation in molecular biology in the UK in the 1950s, right? So why did all of this stuff happen at these times in history? And so there's a great book about this for anybody who's interested called The Eighth Day, which
Okay.
is about the history of molecular biology in 1950s England and the kinds of personalities and how they approach science that I think was kind of a necessary ingredient to science. So that's a book that I think is an amazing book that I would recommend.
And you study a lot of biological systems and a lot of diseases, in fact, with these microscopes. People can do that. So what do you think is the implication of that research? Not at a fundamental level, but an applied level.
Yeah, I mean, I think that many of these diseases, if you really want to understand what's going wrong and how to fix them, these are molecular diseases that affect individual cells in organisms. And so it's hard to get a sense in humans
Right.
as to exactly what is going wrong, right? I mean, the goal is to, in an applied sense, eventually treat a human. But in order to get there, the insights often come from these much simpler organisms.
Mm.
In the worm that I study, Nobel Prizes have been given for the discovery of mechanisms that influence cancer or neurodegenerative diseases.
Yeah.
And these discoveries were first made using very simple microscopes.
Mm.
So I think there is, and that's actually how many microscopes are used today, right? Both mine and others. A cell biologist,
Yeah.
tissue biologist might have some disease model, and they want to understand at the small length scale what exactly is going wrong in a mutant model where a
Right.
gene is overexpressed or something, or how exactly does a drug affect a single cell and heal it, so to
Yeah.
speak, right?
Yeah.
So if you really want to get down to the molecular details, microscopes are invaluable tools. They're a part of the solution.
Over your career, what's one unexpected life lesson that science has taught you?
I think one thing that I've learned is to never we have to kind of, it's natural for us to make assumptions about other people, right? Because we look at CVs, we look at their background.
Yeah.
There's this idea of a pedigree, but that really has nothing to do with somebody's potential for scientific research. And that's something that I've learned time and time again, because I'm fortunate to have had people from state schools, from premier institutions, let's say in China, and there's very little correlation between where they came from and the quality of their thinking. Right. So that's maybe one thing that I myself, like, I know it intellectually, but I have to keep learning that in my heart over and over again, right? To check the assumptions at the door. People have different strengths and weaknesses. And that's like one of the wonderful things about working in research, right? That you can meet many different kinds of people.
Right. Yeah, absolutely. So, right now we see that telescopes are something that, many big scientists have used in observatories, but now they have made a place into your home, because there are a lot of toys which are telescopes. I mean, might not have the greatest quality, but still people use that to actually look at the moon, look at Jupiter, etc., etc. So it's a very commercialized area right now, where people are actually selling it as a commercial toy. So do you see microscopes being sold as that?
To some extent, you can already get these kinds of things off Amazon, right? So you can buy, a cheap microscope, anywhere from $10 to $100. Of course, there's the fold scope of Manu Prakash, which is another thing. And I think the problem is not so much, that you can't get these things, but rather, it's sort of perhaps a problem of like curiosity, right? So you can buy these things, but in my experience, there's sort of like a gee whiz factor when a kid gets this, and then it goes unused for a while. And so the question is, how do you maintain that curiosity? I don't see that so much of a problem of accessibility of the tool as it is as just finding interesting things to marvel at. Right. If you're a kid, or how to nurture that, at home, basically.
Yeah. Okay, so now the last question: what is the message that you would like to give to a young person who would like to explore the microworld?
I think it's a great time to be a microscopist. And the wonderful thing about exploring the microworld is that there's plenty of room. So, if you decide to go into science, if microscopy is a large part of your world, or it's a tool, chances are very, very good you will see something that nobody has ever seen before. So let that be hopefully an inspiration to you to use microscopes in your work.
Okay.
Good luck.
Okay. Thank you so much, Hari, for sharing all your life and experience with us. I think it's a great learning experience, just not for me, but all those who are watching this podcast.
Thanks so much. It was really a pleasure to participate. Thank you.